Sakhalin Kwanzi, who couldn't come and give this talk, but I mean, I'm a co-PI on this grant, so I mean, I think, I can do this presentation for her. So basically, we use a blue water for multi-scale in silica pipeline for predictive safety pharmacology. We actually, like in terms of blue water resources in particular, we use it for molecular simulations of ion channel interactions with drug molecules. And the whole idea of of the pipeline is to start from the level of a protein and drug molecule and go all the way to the level of channel and cell and tissue and organ scale to try to predict what kind of interactions between these ion channels and drug molecule can cause some harm. And I will talk more in detail about that just in a minute. So basically, this is Kalin, who is our team leader, and we have a few other people, like collaborators, Professor Yarofia Avoy, who is in our department, and then external collaborators, Sergei Noskov in Calgary, and Toby Allen at RMIT University in Australia. We have a team of great grad students and postdocs who help us a lot, and for this particular project, it's like Kevin DeMarca and John Dawson who did most of the work, which I'm going to present today. So basically, what we care about, why it matters. Because we are talking about the cardiac arrhythmia, which is like irregular, too slow, or too fast heart rhythm. And it's actually very widespread, so it affects millions of people worldwide, to like 4 million in the US alone. And it's a leading cause of the sudden cardiac death and 50% of cardiovascular, 15% of all deaths, globally, and up to like 350,000 deaths per year here in the US. So basically, I mean, as you probably know, they can be detected at ECG. So this is a normal heart rate. And this is a type of very deadly arrhythmia called tardive point. So, and this tardive point is often the primary cause of this is something called long QT syndrome. And this is like in the cardiogram, you have different QRST, like different, this correspond to different electrical activity in chambers of the heart. And if this interval between this wave, Q and T wave, is prolonged, it's called long QT syndrome. And it's one of the most prominent pro-arrhythmia markers. It could be due to some mutations, or it could be due to unwanted drug interactions. And I will be talking today about the later. Basically, one of the culprits here is this ion channels, and the previous speaker gave like great introductions about ion channels. So the ion channel we're talking about is called hERG, and the name is, it's a voltage-gated ion channel, and so it's basically deactivated by membrane voltage. And it belongs to EAG family, and this is like, EAG stands for Go gene. So this name is comes from exotic dancers, and the fruit flies, which have defect in these genes, they shake legs as those dancers, so that's the name. But the reason why we're interested, so this ion channel is very important of driving the membrane potential down. It's something called repolarization. And if we block this current, so this repolarization is going to slow down, and then we have this something called long QT syndrome. So basically, the main problem is that there are a lot of drugs which can cause this. So some of the drugs are like anti-arrhythmic drugs, like sotalol, and there was some trial to see how how they can help to treat arrhythmia, but the trials had to be stopped because they caused more deaths than placebo. So bad they were. And then there are some drugs which are supposed to be good gastrointestinal agents, but they also cause arrhythmias due to interaction with this hERG channel. So they had to be withdrawn from the market. So actually up to 3% of all prescription drugs worldwide can cause this type of arrhythmias. And now FDA mandates that all of these new drugs in the development and going into the marketplace have to be checked for hERG block and QT signal no prolongation. So, and the failing this test is #1 for drug withdrawal from the market. So, but the main problem here is that not all drugs which block hERG cause arrhythmia. Some drugs, like sotalol I mentioned, it does. But then there are some other drugs which also block hERG and cause some QT prolongation.